So our last speaker this morning is Nancy Laterreur from the University of Sherbrooke in Quebec, who's going to talk about telomerase and RNase interaction.
So hello everybody. So I hope I will keep you from going to lunch, being the last one before I'll try to be quick but interesting. So today I will present you our most recent discoveries in the Wellinger lab about the telomerase enzyme. So as you may know, telomerase is a ribonucleoprotein that is responsible to elongate short chromosome ends at each cell division. It is composed of a large RNA subunit that will help the scaffold of many other proteins. And in Saccharomyces cerevisiae, this RNA is called TLC1, and the proteins that will bind to it is, the main one is S2, which is the catalytic subunit that will use the RNA template to reverse transcribe into DNA, telomeric DNA. Other protein subunits are also essential in yeast, such as S1 and S3, but as I said, the main focus of my talk will be the RNA molecule. So this very long RNA molecule that is called TLC1 is part of the snRNA family. It has a 5' TMG cap, and it is bound by the SM heptamer. This is a very structured RNA, and we can subdivide it into subdomains that are such as the catalytic domain and the recruitment domain. But what is very striking with this RNA is that it is very long, highly structured, but up to now there are only a few proteins that are known to bind to it. So the purpose of my project was to find new partners to the yeast telomerase so that we can rejuvenate this 15-year-old picture. To do so, we performed a purification scheme using MS2 tag within the TLC1 RNA in order to pull down all the proteins that are attached to it via MS2 Pro-A construct. To follow the purification scheme, we also tagged one of the telomerase protein, the S1, with a MIC tag, and we made sure that it was present at each step of the purification scheme. So here are the mass spec results. As you can see, the positive control is among the top hits, and we also found telomerase proteins in that list. But what we also found is three new proteins, POP1, POP6, and POP7, that could be new partners for telomerase. So what are these proteins? They are part of the essential yeast RNase P and MRP complexes. They serve to process precursor RNAs such as tRNA, ribosomal RNA, and tRNAs. And they also scaffold onto RNAs. And for yeast MRP is the NME1 RNA, and for yeast RNase P is the RPR1 RNA. But up until now, there was nothing to do with telomerase for the RNase MRP and RNase P. So we first tried to verify if the POP1, POP6, and POP7 proteins could interact directly with the telomerase RNA. To do so, we used a yeast strain in which the POP1, POP6, and POP7 were tap-tagged, and following IP with those tap-tags, we could analyze RNA through southern blot analysis. So here is our positive control, in which we use the Pro-A S2 version. S2, as I said, is the catalytic subunit of telomerase, so it serves as a positive control for TLC1 pull-down, but not for the NME1 RNA, which is part of the RNase MRP complex. As you can see on the northern blot here, all three POP6, POP7, and POP1 all co-IP TLC1 RNA together with the NME1 RNA, which is part of their normal complex. One of the other members of the RNase MRP, POP4, and a few others that we tested did not co-IP TLC1. This suggests that POP1, POP6, and POP7 interact specifically with telomerase in that case. We also wanted to test if these IPs were active for telomerase. So using a modified primary extension after reverse transcription by telomerase, we could observe on a polyacrylamide gel that all three POP6 and POP7 and POP1 could co-IP active telomerase particles. As you can see on this polyacrylamide gel, we can see the telomerase elongation products. It has been described in the literature that the POP1, POP6, POP7 proteins can recognize a specific part of the NME1 and RPR1 RNA that is called the P3 domain. This stem loop structure is essential for the enzymes to function. Looking at the sequence and structure of TLC1, we also found a similar P3 domain that may be implicated in the POP6, POP7 binding. And as you can see with the blue circles here, most of the nucleotides that are identical in these three P3 domains have been described to be the exact binding site for POP6 and POP7 in the RNase context. But in a study from our lab in the previous years, we also found that this region was essential for telomerase function, but we did not know why at that time. So we did a swapping experiment in which we swapped the potential P3 domain of TLC1 with the P3 domains of either NME1 or RPR1 RNase. And in yeast, what we can do to analyze telomerase function is to do telomerase length analysis. And through southern blotting, after digesting the DNA with an enzyme that will liberate the chromosome ends, we can see telomere sequences on the southern blot with a telomere-specific probe. So as you can see here, wild-type telomere length is about 1.2 kb in length, but when we remove one of the components of telomerase, so for here it's TLC1, we have a shortening of telomeres and a telomerase null pattern appearing. When we remove the potential P3 domain of TLC1, we also have a telomerase null phenotype. But as soon as we swap back either the NME1 RNA P3 domain or the RPR1 P3 domain, we restore the telomere length back to wild type. In order to see if this potential P3 domain is a functional P3 domain in the context of the RNase activity, we did the reverse swapping experiment in which we took the potential P3 domain of TLC1 and put it into the NME1's P3 domain in place of the NME1 P3 domain. And as you may remember, I told you that the NME1 is essential for yeast to grow. And when we put the P3 domain of TLC1 into the NME1 RNA, we restore the growth of the yeasts. So this suggests that TLC1 has a fully functional P3 domain that will function in the context of the RNase MRP. In order to test if the POP6, POP7 binding is via this P3 domain for TLC1, we used a strain in which we expressed two types of TLC1. So the first copy was the endogenous wild-type version, and we also co-expressed a longer version which has the MS2 tags at the end. So when we analyze after IP with the POP6 step, or we also did it for POP7, we can observe both forms of TLC1 after the co-IP. But when we remove the P3 domain in TLC1, we see that the TLC1 MS2 version, which is the mutated form here, is lost in the northern blot analysis. On the protein side, we wanted to verify that if upon deletion of the P3 domain in TLC1, we lose this interaction between the POP6, POP7, and the RNA. So we tagged one of the SM proteins that is bound to TLC1 with a MIC tag. And following the co-IP with anti-MIC antibody, we could observe that upon deletion of the P3 domain, we have a reduction in the binding of both POP6 and POP7 in our co-IP. We also wanted to verify if the P3 domain was essential for telomerase integrity. So using the same kind of principle, we tagged one of the SM protein with the HA tag this time. And using the S1 MIC and S2 MIC construct, we've been able to show that removal of the P3 domain in TLC1 decreased dramatically the amount of S1 and S2 in our co-IP fractions. So this suggests that the P3 domain is required to either complex assembly or complex stability. And finally, since we think that this binding is important for telomerase integrity or stability, it should be that if you reconstitute this activity in vitro and add the recombinant proteins to the system, you can stimulate telomerase activity. So this is what we did. Using an RRL system in which we co-express a shorter version of TLC1, together with a recombinant pro-AS2, we can reconstitute telomerase activity in vitro. So to this system, we added recombinant POP proteins, and we can observe on this polyacrylamide gel that this addition stimulates telomerase activity, especially when we add the POP1 protein. So in conclusion, I hope I convinced you that we found 3 new proteins that are part of the telomerase complex. So these proteins are named POP1, POP6, and POP7. They bind to telomerase via a very specific domain that is called the P3 domain, and that is shared between the RNase P and MRP complexes. This P3 domain seems essential for telomerase integrity, as it may stabilize the complex or help in its assembly. So you can read the complete story in our most recent publication. And with that, I would like to thank all the members of the lab, especially Bruno Lemieux, who did the work with me on this. So Sherbrooke is situated north of the wall from here in Canada. And I would like to take any questions you may have. No questions.
Is there a question up there? All right, I think maybe everyone's anxious for lunch. So maybe with that, we'll thank the speakers and everyone for your attention.